Mineral nutrients, Chapter 2, NCERT. The basic needs of all living organisms are essentially the same. They require macromolecules such as carbohydrates, proteins, and fats, and water and minerals for their growth and development. This chapter focuses mainly on inorganic plant nutrients, wherein we will study the methods to identify elements essential to growth and development of plants and criteria of establishing this essentiality. We will also study the role of essential elements, their major deficiency symptoms, and mechanism of absorption of these essential elements. The chapter also introduces you briefly to the significance and mechanism of biological nitrogen fixation. Methods to study the mineral requirement of plants. In 1860, Julius von Sack, a prominent German botanist, demonstrated for the first time the plant could be grown to maturity in a defined nutrient solution in complete absence of soil. This technique of growing plants in a nutrient solution is called hydroponics. Since then, a number of improvised methods have been employed to try and determine the mineral nutrients essential for plants. These essence of all these methods involves the culture of plant in soil-free defined mineral solution. These methods require purified water and mineral nutrient salts. Can you explain why is this so essential? After a series of experiments in which the roots of the plant were immersed in nutrient solution and wherein an element was added, substituted, removed, or given in varied concentration of mineral solution, stable for plant growth was obtained. By this method, essential elements were identified and their deficiency symptoms discovered. Hydroponics has been successfully employed as a technique for the commercial production of vegetables such as tomatoes, cucumber, and lettuce. It must be emphasized that the nutrient solution must be adequately aerated to obtain the optimum growth. What would happen if solution were poorly aerated? The alternative use of the hydroponic technique is given in figure. Alright. So this is for adding water and nutrient solution. Everything's cotton, and this aerating tubes, nutrient solution there. Alright. Now to open to essential mineral elements. Most of the minerals present in soil can enter plants through. In fact, more than 60 elements of the 105 discovered so far are found in different plants. It means out of 105 elements, 60 elements are found in plants. Some plant species accumulate selenium, some other gold, while some plants grow near nuclear test sites take up radioactive strontium. Mineral even a very low concentration of 10 inverse 8 gram per milliliter. 10 inverse 8 gram per milliliter. The question is in very low concentration. Alright, the question is whether the diverse mineral elements present in plants, for example, gold and selenium, as measured ever. Really necessary for plants? How do we decide what is essential for plants and what is not? Criteria for essentiality. Okay, here is the nutrient solution here, pump here. And hydroponics plant production, plant are grown in a tube or through placed on a slight incline. A pump circulates the nutrient solution from the reservoir to elevate it. The solution flows down the tube and return to the reservoir due to gravity. The solution flows down to the reservoir due to gravity. Insect source of plant whose roots are continuously bathed in aerated nutrient solution. The arrows indicates the direction of flow. Criteria for essentiality. The criteria of essentiality of an element are given below. Okay. The element must be absolutely necessary for supporting normal growth and production. In the absence of the element, the plant will not complete their life. Even said the same. Complete the life cycle or set the same. The requirement of the element must be specific. Means life cycle complete. The requirement of the element must be specific and not replaceable by other element. In other words, the deficiency of any one element cannot be met by supplying some other elements. The element must be directly involved in the metabolism of the plant. Based on the above criteria, only few elements have been found to give some essential for plant growth and metabolism. These elements are further divided into two broad categories based on quantitative requirements. That is macromolecules and micromolecules. So what are macromolecules? Let's see. Macromolecules are generally present in plants in a large amount, in excess of 10 millimole kg inverse. You can see it's 10 millimole kg inverse of dry matter. The macromolecules include the carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, potassium, calcium, and magnesium. So the trick to remember this is trick to remember. You can say simply it's CHON corn PK cast mug. Corn PK cast mug. You want to go 5, 6, 7, 8, 9. You can see 1, 2, 3, 4, 5, 6, 7, 8, and 9. Alright. Now of this carbon, hydrogen, oxygen are mainly obtained from CO2 and H2O. Whether CHO can be obtained from the CO2 and H2O, while the other are obtained from the soil as mineral nutrients. Alright, and the rest can be obtained from soil. Now coming to micronutrients. Micronutrients are you also call it trace elements. Alright, so these are the needed in very small amounts. It's less than 10 millimole kg inverse of rice in dry matter. This includes iron, magnesium, copper, molybdenum, zinc, boron, chlorine, and nickel. So the trick to remember is trick for micronutrients will be like fe, ku, mo, ni, man, jan, kal, bit, boron. Alright, 1, 2, 3, 4, 5, 6, 7, 8, 1, 2, 3, 4, 5, 6, 7, 8. So there are 8 micronutrients and 4, 5, 6, 7, 8, 9 macronutrients. So overall there are 17 essential elements. You can see 8 plus 9, 17. Essential elements can also be grouped in 4 broad categories on the basis of their diverse functions. These categories are essential elements as components, biomolecules, and hence structural elements of cell. The carbon, hydrogen, oxygen, and nitrogen. So components of biomolecules and hence structural elements of cells. These are carbon, hydrogen, oxygen, and nitrogen. Alright. So essential elements that are components of energy-related chemical compounds in plants, magnesium and chlorophyll, and phosphorus and ATP. So these are the essential elements related to energy-related essential elements. Okay. Now coming to the 6 elements that activate or inhibit enzymes. For example, magnesium glucose is an activator of glucose, phosphate carboxylate, rubisco, and phosphoenol pyruvate carboxylate. Okay. So rubisco and phosphoenol pyruvate carboxylase activator है magnesium 2 plus ion, number of which are essential elements of oxidative carbon filtration. Zinc plus 2 is an activator of alcohol dehydrogenase. Okay, we know this. Zinc plus 2 is an activator of alcohol dehydrogenase, and molybdenum of nitrogenase, and molybdenum is an activator of nitrogenase. So during nitrogen metabolism, can you name a few more elements that fall in this category? For this, you will need to recollect some of the biochemical pathway you have studied earlier. Now, some essential elements can alter the osmotic potential of cell, like potassium plays an important role in opening and closing stomata by altering the osmotic potential of guard cells. So you may recall the role of minerals as solute in remaining water in cell. Now, coming to the role of macro and micronutrients. So here we can say essential elements perform several functions. They participate in various metabolic processes in plant cell, such as permeability of cell, what are its functions? Permeability of cell membrane, maintenance of osmotic concentration in cell sap, electron transfer system, ETS, buffering action, enzymatic activity, and acts as major constituents of macromolecules and coenzymes. Various forms of functions of essential nutrient element given below. So let's write it once. What are the functions? Permeability of membrane, osmotic maintenance of concentration of cell sap, ETS, permeability, ETS, maintenance of the concentration of cell sap, and buffering action, enzymatic activities, and okay, and act as major constituents of macromolecule and coenzyme. Coenzymes. Now, let's see. Various forms and functions of essential nutrients elements are given below. So let's see one by one. Nitrogen. So it is essential nutrient element required by plant in the greatest amount. Required by plant in the greatest amount. It is observed mainly as NO3, observed in the form of nitrite. Okay. So those some are also taken up as nitrate and ammonium ion. Because mainly it's taken in this form. Those some are also taken in the form of nitrate, sorry, NO2 minus, and ammonium ion, and H4 plus. Right? So nitrogen is required by all parts of the plant, particularly the mesmatic tissue. So this can be required by all parts of the plant, but particularly the mesmatic tissue and metabolically active cells, which ethical reasons you can say metabolically active and mesmatic and okay, so that's required by all parts of the plant. So nitrogen is one of the major constituent of protein, nucleic acid, vitamins, and hormones. So nitrogen is one of the major constituent of protein, nucleic acid, vitamins, and hormones. So come to phosphorus. So phosphorus is observed by the plant from soil. Yeah, as potassium ion. This is phosphorus. It's phosphorus. So phosphorus is observed in plants from soil in the form of phosphate ions, either H2PO4 or HPO4. 2 and 4 is constituents. So it's either H2PO4 minus or HPO4 2 minus. Alright. So phosphorus is a constituent of cell membrane, certain protein, all nucleic acid phosphorus is a constituent of cell membrane, certain proteins, all nucleic acid and nucleotides, and is required for all phosphorylation reactions. So where the phosphorus is required, it's required for the maintenance of cell membrane, it is constituent of cell membrane, and certain proteins or nucleic acid, not all, say certain, it's certain proteins, but all nucleic acid, alright, and nucleotides, and is required for phosphorylation reactions. Alright. Now coming to potassium. So potassium is observed in the form of Kpus in plants and is required in more abundant quantities in the mesmatic tissue. Potassium. It's observed in potassium in plants. It is required in more abundant quantities in mesmatic tissue. Like phosphorus, it's also required in mesmatic tissue and metabolically active cells in birds, leaves, and root tips, apical cells. Mesmatic tissue, buds, leaves, and root tips. Potassium helps to maintain anion-cation balance in cells and involves in protein synthesis, opening and closing of stomata, activation of enzyme, and in the maintenance of rigidity of cells. Okay, so on this protein synthesis. Calcium. Plants of calcium from soil. Where do plants take calcium from? Soil. In the form of calcium ions, calcium ion, Ca+2. Calcium required by mesmatic and differentiating tissue. During cell division, which is used in the synthesis of cell wall. During cell division, it is used in the synthesis of cell wall, particularly as calcium peptide in middle lamella. During cell division, it is used in synthesis of cell wall. During cell division, is used in the synthesis of cell wall, particularly as calcium peptide in middle lamella. It's also needed during the formation of mitotic spindle. It accumulates in older leaves. It's involved in normal functioning of cell membrane. It activates certain enzymes and play an important role in regulating metabolic activities. It activates certain enzymes, an important role in regulating metabolic activities. Coming to magnesium, it is a transfer form of divalent magnesium ion, Mg2+. It activates enzyme of respiration, photosynthesis, and is involved in the synthesis of DNA and RNA. So, DNA synthesis. Alright, so it's not the synthesis of DNA and RNA. Magnesium is a constituent of magnesium is a constituent of ring structure chlorophyll and helps in maintaining the ribosome structure. Okay, so it's also part of the ring of chlorophyll and also helps in maintaining the structure of ribosomes. I think it's to to join the two subunits of ribosomes. Magnesium is required in 0.0001 mg. Now come to sulfur. The sulfur contains sulfur from sulfate and SO4 2 minus. Sulfur is a sulfate. Okay, sulfur is present in two amino acids, cysteine and methionine. Sulfur is present in two amino acids, cysteine and methionine, and is main constituent of several coenzyme. Also, it's constituent of several coenzymes and vitamins like thiamine, biotin, coenzyme A, and production. Alright, so it is main constituent of several coenzymes and vitamins. What vitamins are there? Thiamine, thiamine, biotin, coenzyme A, and pyridoxine. Plant obtain iron. Plant obtain iron from ferric ions Fe3+. It's required in larger amounts in comparison to other It's required in larger amount in comparison to other micronutrients. It is an important constituent of protein involved in transfer of important constituents of protein involved in transfer of electron in ETS, like pyridoxine in cytochromes. Plants obtain iron in the form of ferric ions. It's required in larger amounts in comparison to other micronutrients. It is an important constituent of protein involved in transfer of electrons, like pyridoxine and cytochromes. It is reversibly oxidized from Fe2+ to Fe3+. And iron form of ferric ions. It's quite a large amount in comparison to micronutrient. It will constitute of protein involved in transfer of electrons, pyridoxine, cytochrome. It's reversibly oxidized from Fe2+ to electron transfer. It activates catalase enzyme. It is essential for formation of chlorophyll. Also essential for formation of chlorophyll. Now coming to the manganese. Alright. Coming to manganese. It is observed in the form of manganese ions, plus 2. It activates many enzymes involved in photosynthesis, respiration, and nitrogen metabolism. The best defined function of manganese is in the splitting of water to liberate oxygen during photosynthesis. Alright, so it also activates many enzymes involved in photosynthesis, respiration, and nitrogen metabolism. Alright, the best defined function of manganese is the splitting of water to liberate oxygen. Manganese is the splitting of water, splitting, and during photosynthesis to liberate oxygen. Alright. Now coming to zinc. It activates an enzyme called carboxylase and obtained in the form of ZN2+ ion. It's also needed in synthesis of oxygen. Come to copper. It's observed with cupric ion, Cu2+. It's essential for overall metabolism in plants like iron. It's associated with certain enzymes involved in redox reaction and is it's essential for overall metabolism of plants like iron. It's associated with certain enzyme involved in redox reactions and is reversibly oxidized from Cu+ to Cu2+, iron Cu+ to Cu2+. Okay, it is reversibly oxidized. Now boron. It is observed as BO3, 3- and BO4. You have to remember this because the questions are directly asked from the line of NCERT. BO3, 3- or B4, 7, 2-. Boron is required for uptake and utilization of calcium and membrane functioning, boron germination, cell elongation, cell differentiation, and carbohydrate translocation. So there are many functions of boron mentioned here. You will remember all. Alright, so let's write it once. So for the uptake and you can see utilization of calcium ion, cell differentiation, cell elongation, carbohydrate translocation, and pollen germination, membrane functioning, Now chlorine. Okay, molybdenum. Plants obtain it in the form of MOO2-. Molybdenum O2 plus. Okay, it's a component of several enzymes, including nitrogenase and nitrate reductase, both of which participate in nitrogen metabolism. It is a component of several enzymes, including nitrogenase and nitrate reductase, both of which participate in nitrogen metabolism. So, nitrogenase and nitrate reductase participate in nitrogen metabolism. Chlorine, observed in the form of chlorine ion. along with sodium and potassium ion. It helps in determining the solute concentration, and the anion balance helps in determining solute anion balance in cell. It is essential for water splitting reactions in photosynthesis, a reaction that leads to oxygen evolution. It is essential for the water splitting reaction in photosynthesis, a reaction that leads to oxygen evolution. Okay. It is essential for water splitting reaction in photosynthesis, a reaction that leads to oxygen evolution. Also, along with chlorine, also manganese helps in splitting of water, right? Yeah, splitting of water. So manganese and chlorine helps in splitting of water and helps in evolving oxygen. Alright, now coming to deficiency symptoms of essential elements. So you can see, whenever the supply of essential elements become limited, plant growth is retarded. The concentration of essential elements below which plant growth is retarded is termed as critical concentration. You can see the concentration of essential elements below which plant growth is retarded is termed as critical concentration. Now the element is said to be deficient when present below the critical concentration. When it's present below the critical concentration, it's called then the particular element is called deficient. Okay? Now, since each element has one or more specific structure or functional role in plants, in absence of any particular element, plants show certain morphological changes. These morphological changes are indicated of the certain element deficiency and co-deficiency symptoms. Alright, the deficiency symptoms vary from element to element, and they disappear when the deficient mineral is provided to the plant. However, if deprivation continues, it may eventually lead to the death of the plant. The parts of the plant that show the deficient symptoms also depends on the mobility of the element in the plant. Okay, so deficient symptoms also depends upon the mobility of the element in the plant. For elements that are actively mobilized within the plants and exported to young developing tissue, the deficiency symptom tends to appear first in older tissue. You can see. For elements that are actively mobilized within the plant and exported to young delivering tissue. That's why the deficiency symptoms first appear in older tissue. For example, the deficiency symptoms of nitrogen, potassium, magnesium are visible first in senescent leaves. Nitrogen, potassium, and magnesium first observed in the senescent old leaves because it has been transports to young leaves. In older leaves, biomolecules containing these elements are broken down, making these elements available for mobilizing to younger leaves. Okay, the deficiency symptoms tend to appear first in young tissue wherever the elements are relatively immobile. For immobile elements, the deficiency symptoms are appears in young leaves or tissues, because it's obviously it's immobile for immobile, but and are not transported out of the mature organs. For example, sulfur and calcium. For elements like sulfur and calcium, these sulfur and calcium are immobile, so its deficiency symptoms will first appear in young tissue, while nitrogen, potassium, and magnesium are mobile, so its symptoms will first appear in old tissue. That's all part of structural elements. This is my part of structural component of the cell, and hence are not easily released. These aspects of mineral nutrients of plant is of great significance and importance of agriculture and horticulture. It has great significance in the field of agriculture and horticulture. Alright, the kind of deficiency symptoms of plants includes. So these are the names and kinds of chlorosis, necrosis, stunted plant growth, premature fall of leaves and buds, and inhibition of cell division. Also inhibits the cell division. Right. So what chlorosis is Now, chlorosis is basically the loss of chlorophyll leading to the yellowing of leaves. Necrosis mainly death of tissue, particularly leaf tissue. It is due to the deficiency of death of tissue, particularly leaf tissue. And inhibition of cell division. It's a cause of molybdenum, sulfur, potassium, and nitrogen. Alright, so premature leaf fall. You know that. Also delay in flowering due to molybdenum, sulfur, and nitrogen. Yellowing chlorosis is due to necrosis due to kaku kamak. It's due to kaku kamak. Okay, you can remember like this. And now chlorosis. Chlorosis is due to. Yeah, I'm writing FK SN MN ZN MGMO. These are the elements responsible for the chlorosis. It's a fake sun manjan magma. Like this, remember FKSN MN ZN MGMO. Now let's inhibition of cell division is due to MOSKN, Muskan, you can remember like this, and delay in flowering due to MOSM, or you can say MOSM, MOSN, and death of tissue, particularly the leaf tissue, it's kaku kamak. Alright. Now chlorosis also chlorophyll into yellowing of leaf, yellowing leaf. This is due to deficiency of these elements already written. And likewise necrosis or death of tissue, particularly leaf tissue, is due to deficiency of kaku kamak. Lack or low level of MOSK and Muskan, you can say, cause an inhibition of cell division. And some elements like MOSN delay in flowering if their concentration in plants is low. Okay. You can see from the above chart, the deficiency of any element can cause multiple symptoms, and that the same symptoms may be caused by deficiency of one of the several elements. Hence, to identify the deficiency elements, one has to study all the symptoms. Alright, you can see. That the deficiency of any element can cause multiple symptoms, and the same symptoms can be caused by deficiency of one of the several elements. Hence, to identify the deficient element, one has to study all the symptoms developed in all the various plants, parts of the plants, and compare them with the available standard tables. We must also be aware that different plants also respond differently to the deficiency of same elements. Now, coming to the toxicity of micronutrients, the requirement of micronutrients is always in low amount, while their moderate decrease causes deficiency symptoms and moderate increase cause toxicity. In other words, there is a narrow range of concentration at which the elements are optimum, and mineral ion concentration in tissue that reduces the dry blood pressure by about 10% considered as toxic. Since critical concentration are widely among different micronutrients, the toxicity symptoms are difficult to identify. Toxicity levels for any element is also vary for different plants. Many times, excess of an element may inhibit the uptake of another element. Many times, excess of an element may inhibit the uptake of another element. For example, the prominent symptoms of manganese toxicity is appearance of brown spot surrounded by chlorotic veins. Right. Symptoms of manganese toxicity is brown spots surrounded by chlorotic veins. It's important to know that manganese competes with iron and magnesium. So manganese competes with iron and magnesium for uptake. And with magnesium and with manganese for binding with enzymes. For uptake and with manganese for binding with enzymes. Manganese also inhibits. Okay, so iron and magnesium for uptake, and manganese for binding. Manganese also inhibits calcium translocation in pseudopeps. Therefore, excess of manganese may in fact induce deficiency of iron, magnesium, and calcium. Calcium translocation in pseudopeps. Calcium. This what happens as symptoms of manganese toxicity may actually be deficiency symptoms of iron, magnesium, calcium. Can this knowledge be of some importance to a farmer, a absorption or even for you in your kitchen garden? Mechanism of absorption of elements. Much of the studies on mechanism of absorption of elements by plants has been carried out in isolated cells. These studies reveal that the process of absorption can be demarcated into two main phases. In the first phase, an initial uptake of ions into the free space or outer space in cell, and the apoplast is passive. And the second phase is uptake of ions and the apoplast is passive. The second phase is uptake the ions and second phase of uptake. The ions are taken slowly into the inner space. The symplast of the cell. Outer space, free space or outer space called apoplast. Inner space is called symplast. It's slow, it's rapid. Alright, you can see here about apoplast. It's rapid, fast, and free space, or you can say outer space. It's passive. Now symplast, it's slow, it's inner space, and it's active. The passive movement of ions in apoplast usually occurs through ion channels in the transmembrane protein. In the transmembrane proteins that function as selective pores. Transmembrane proteins that function as selective pores. Plus movements of ions into the apoplast usually occur through ion channels. The transmembrane proteins that function as selective pores. On the other hand, the entry or exit of ions from the symplast required expenditure of metabolic energy, which is an active process. The movement of ions is usually called flux. The inward movement is the inward movement to the cell is influx, and outward movement is efflux. Here we read the spectrum of micronutrients uptake and translocation chapter level. Okay. Translocation of solutes. Minerals are translocated through xylem along with the ascending stream of water, which is pulled up through the plants by transpirational pull. Analysis of xylem sap shows the presence of mineral salts in it. Use of radioisotopes of mineral elements also sustain the view that they are transported through xylem. Soil as a reservoir of essential elements. The majority of the nutrients are essential for the growth and development of plants from available to the roots due to withering and breakdown of rocks. These processes enrich the soil with dissolved ions and inorganic salts. Since they are derived from the rock minerals, the role of plant nutrient is referred to as mineral nutrition. Soil consists of a wide variety of substances. Soil is not only supplied minerals, but also harbors nitrogen-fixing bacteria. Other microbes hold water supplies to the roots and act as matrix that stabilizes the plant. So not only supplies minerals, but also harbors nitrogen-fixing bacteria. Other microbes hold water supplies air to the roots and act as matrix for the matrix that stabilizes the plant. Since deficiency of essential minerals affect the crop yield, there is often a need of supplying them to fertilizers. Both the macro and micronutrients form components of fertilizer are supplied as per need. Now coming to the metabolism of nitrogen. The first nitrogen cycle. Apart from carbon, hydrogen, and oxygen, nitrogen is the most prevalent element in living organisms. Nitrogen is constituent of amino acid protein, hormones, chlorophyll, and many of the vitamins. Amino acid protein, hormones, chlorophyll, and many of the vitamins. Okay. Plants compete with microbes for the limited nitrogen that's available in soil. Here is the nitrogen cycle. Right. 4 pages are left. Here you can see nitrogen cycle, how it works. You can see atmospheric nitrogen. So it's biological. There are 3 ways of fixing nitrogen. It's biological nitrogen fixation, biological and fixation, industrial, electrical, and through biological and industrial, it's fixed in the form of NH3 ion, ammonium, and which is further converted to nitrate, then soil in nitrogen pool, and then into nitrite. Okay, so nitrite is also fixed in the form of by process of denitrification, or we can say nitrogen fixed by denitrification and converted in nitrate. Now ammonification, ammonia, and which is okay. So nitrite for the taken with plant biomass, or which is further plant biomass taken by animal biomass, or when it plant get animal get it's further converted to ammonia with the process for ammonification, and here it's ammonia forms. Alright, so it's a cycle, going cycle. The nitrogen cycle showing reason there were 3 main nitrogen pools, 3 main nitrogen atmospheric, soil, and biomass. So I think question can be asked in part of the 3 nitrogen pools. You can say atmospheric, 3 main nitrogen pools, atmospheric, soil, and biomass. Alright, does nitrogen limited nutrient for both natural and agricultural ecosystem? You can say nitrogen limited nutrient for both natural and agricultural ecosystem. Nitrogen exists as 2 nitrogen atoms joined by a very strong triple covalent bond. And 2. The process of conversion of nitrogen to ammonia is termed as nitrogen fixation. In nature, lightning and ultraviolet radiation provide enough energy to convert nitrogen to nitrogen oxides. Lightning and ultraviolet radiation provide nitrogen to NO2 and N2. NO, nitrogen oxides, NO2 and N2. Okay, so nitrogen dioxide, dinitrate oxide, something. Alright, so dinitrogen oxide. Alright, so industrial combustion, forest fire, automobile exhaust, and power generating stations are also source of atmospheric nitrogen oxides. Industrial combustion, forest fires, automobile exhausts, and power generating stations are also source of atmospheric nitrogen oxides. Decomposition of organic nitrogen of dead plants and animals, ammonia modification. Some of these ammonia volatilizes and re-enters the atmosphere, but most of it is converted into nitrate by soil bacteria in the following step. So these are the steps. Ammonia, how ammonia is converted to nitrite. So let's write the step once. It's up to NH3 and supplied oxygen gets converted to NH2 minus S2H plus S2H2. Alright, now again then 2 NH2 minus converted into, when supplied oxygen gets converted into nitrite. Alright, ammonia is first oxidized to nitrite by bacteria, nitrosomas or nitrococcus. The nitrite is further oxidized to nitrate with the help of nitrobacter. I see. The nitrite is oxidized to it's nitrite, and it's nitrate with the help of bacterium nitrobacter. Alright, so nitrocystis. See? Nitrosomonas and nitrococcus. And here bacteria required is nitrobacter and nitrocystis. These steps are called nitrification. These nitrifying bacteria are called chemoautotrophs. Nitrate is formed in absorbable plants and transported through the leaves. In these, it is reduced to form ammonia, and finally forms the amine group of amino acid. Amino acid. Nitrate present in soil is also reduced to nitrogen by the process of denitrification. Very few living organisms can utilize nitrogen in the form of N2. Very few living organisms can utilize the nitrogen in the form of N2 abundantly in the air. Only certain prokaryotic species are capable of fixing nitrogen. Reduction of nitrogen to ammonia by living organism is called biological nitrogen of fixation. The enzyme nitrogenase, which is capable of nitrogen reduction, is present exclusively in prokaryotes. Such microbes are called N2 fixers. The nitrogen fixing microbes could be free or symbiotic. Example of free nitrogen fixing anaerobic bacteria are Azotobacter and Vaginicia, while Rhodiosporidium is anaerobic and free living. Aerobic free living are it's classified. So see, this takes last piece. So getting a piece. Okay, so here it is. N2 fixers mean free living. You see, aerobic. So aerobic are Azotobacter, Vaginicia, anaerobic. We are talking about bacteria. So anaerobic is what? Rhodiosporidium. And Clostridium. Also some bacteria that help in nitrogen fixations are Bacillus. It's also free living. Also some cyanobacteria helps. Free living are Anabena, Oscillatoria, Unicera and cylindric spermum. And symbiotic is symbiotic cycle. Here also you can find Nostoc and Anemone. Basically in Cycas and Ajola, in Cycas and some lichens. Hope it helps. Symbiotic nitrogen nitrogen fixing microbes could be free living or symbiotic. Examples of free living nitrogen fixing aerobic microbes and Azotobacter and Vaginicia, while Rhodiosporidium is anaerobic and free living in addition to a number of bacteria such as Anabena and Nostoc are also free living nitrogen fixers. The symbiotic biological nitrogen fixation. Several types of symbiotic biological nitrogen fixing associations are known. The most prominent among them is legume bacteria relationship. Species of Rhodes rhizobium has such relations with the roots of several legumes such as alfalfa, sweet clover, sweet pea, lentils, garden pea, broad bean, clover beans, etc. So Fabaceae family, so it's associated with Fabaceae family, and it has symbiotic relations with these plants named alfalfa, sweet clover, sweet pea, lentils, garden pea, broad bean, clover bean. And the most common association on root is nodule. These nodules are small or growth on root. The microphangia also produce nitrogen fixing nodules in the root of non-legume plants, example ulnus. Okay, both rhizobium and phangia are free living in soil, but symbionts. Free living symbionts. Atmospheric nitrogen strength. Okay, now approved any one plant of a common pulse just before flowering, you'll see a spherical upgrowth on the root. These are nodules. If you cut through them, you'll notice that central portion is red or pink. What makes the nodule pink? This is due to the presence of igneous hemoglobin or lake hemoglobin. This is a method of the formation of nodule. Nodule formation involves a sequence of multiple interaction between rhizobium and root of the host plant. Principal stage of the nodule formation is summarized as follows: rhizobium multiply and colonize the surrounding roots and get attached to epidermal and root cell. The root cell curl and the bacteria invade the root hair. An infection thread is produced carrying the bacteria into an infection thread is produced carrying the bacteria into the cortex of root, where they initiate the nodule formation in the cortex of root. So where the nodule formation is initiated in the cortex of root. So then then the bacteria are released from thread into the cell, which leads to a differentiation and specialized, which leads to the differentiation of a specialized nitrogen fixing cell. Then the bacteria are released from the thread into the cells, which leads to a differentiation of specialized nitrogen fixing cells. The bacteria are released from the thread into the bacteria released from the thread into the cell cortex cells, which lead to differentiation of specialized nitrogen fixing cells. The nodule thus formed established a direct vascular connection with the host for exchange of nutrients. The nodule thus formed established a direct vascular connection with the host for exchange of which. So the nodule. Okay. The nodule thus formed established a direct vascular connection with the host for exchange of nutrients. It establishes a direct vascular connection with host for exchange of nutrients. These events are depicted in figure 2.4. The nodule connects all is necessary. Okay. The nodule contains all necessary biochemical components such as enzyme, nitrogenase, and hemoglobin. The enzyme nitrogenase is amorphic protein and catalyzes the conversion of phosphate nitrogen and ammonia. The first stable product of nitrogen fixation. Ammonia, which is the first stable product of nitrogen fixation. Which here you can see soil particles, root hair, bacteria are present near the root hair. So when bacteria start multiplying, the root hair curls and forms a hook-like structure, which further develops an infection thread containing bacteria, and it reaches to the inner cortex and pericycle under division. So thus the mature nodules are formed. Development of root nodules in soybean. Okay. So it's shown in fourth soybean plant. The rhizobium bacteria is susceptible. Root hair divides near it. The successful infection to the root hair causes it to curl. Infected thread carries the bacteria in inner cortex plaza. The bacteria modified into roadside bacteroids. Bacteroids is here referred to the rhizobium bacteria. Okay, and cause the inner cortical and pericycle cells to divide. Division and growth of cortical and pericycle cells lead to nodule formation. A mature nodule is mature nodule is split. Bacteria modified into roadside bacteroids and cause inner cortical and pericycle cells to divide. Division and growth of cortical and pericycle cells lead to the nodule formation. A mature nodule is complete with vascular tissue, continuous with those of the root. And the reaction is as follows: N2 H electron, 16 ATP. Okay, for 2 nitro, for N2 it's 16 ATP into ammonia is produced. For 1 ammonia, required ATP will be 8. And 16 inorganic phosphate. Alright, it's for the 2 NH3. The enzyme nitrogenase is highly sensitive to the molecular oxygen. It requires anaerobic condition. The nodule have adaptations that ensures the enzyme is protected from oxygen, that's like hemoglobin that surrounds around the nodule and that blocks the path of oxygen and to make the condition anaerobic. Okay, to protect these enzymes, the nodule contain oxygen is scavenger called like hemoglobin. It's interesting to note that the microbes live as aerobes under free living condition where nitrogenase is not operational, but during nitrogen fixation they become the anaerobic. Let's protect the nitrogenase enzyme. It's strange. You must have noticed that aerobic reaction that ammonia synthesis by nitrogenase requires a substrate. Its enzyme nitrogenase. It's binding of substrate. Okay, that's a reduction. 2H hydrogen supplied. Hydrogen is added. Again, hydrogen is added. Further reduction takes place. Now, free nitrogenase can bind to another molecule of N2 and release the products. You can see. This is how ammonia is produced, or you can say fixed by the nitrogenase. The steps of conversion of atmospheric nitrogen to ammonia by nitrogenase enzyme complex found in nitrogen fixing body. Very high input of energy, 8 ATP for each NH3 produced. The energy required is obtained from the respiration of host cell. Is obtained from the respiration of host cell. Now, fate of ammonia. At physiological pH, physiological means normal. The ammonia is protonated to form NH4 ammonia. It's protonated. We show that 2H plus hydrogen was added there and reduction took place. While most of the plants can assimilate nitrate as well as ammonia ions, the latter is quite toxic to plant. They are quite toxic to plants and hence cannot accumulate in them. Let's now see how the ammonia is used to synthesize ammonia. amino acid in plants. There are two main ways in which these can express it: reductive amination and transamination. In this process, in reductive amination, ammonia reacts with alpha-ketoglutarate acid and forms glutamic acid, as indicated in the equation. Alpha-ketoglutarate acid can provide reactive ammonia in the presence of NADPH and enzyme glutamate dehydrogenase is required. It's later formation of glutamic acid with hydrogen and NADP. NADP is oxidized here, and its acid is reduced. Now come to transamination. It involves the transfer of amino acid from one amino acid to keto group. It involves the transfer of amino group from one amino acid to keto group of keto acid. Transfer of amino group. Transfer of amino group from one amino acid to the keto group of keto acid. Glutamic acid is the main amino acid from which the transfer of ammonia. Glutamic acid is the main amino acid from which the transfer of ammonia. The amino group takes place, and other amino acids are formed due to transamination. When the transfer of ammonia amino group takes place, the other amino acids are formed through transamination. The enzyme transaminase catalyzes all such reactions. It involves transfer of amino group from one amino acid to keto group of keto acid. Correct? Glutamic acid is the main amino acid from which the transfer of ammonia amino group takes place, and other amino acids are formed through transamination. The enzyme transaminase catalyzes all such reaction. Through transamination. Amino donor. This one is amino acceptor. Okay. You can say amino group transfer. The two most important amides, asparagine and glutamine, found in plant structural part of protein. Asparagine and glutamine found in plant structural part of protein. They are formed from two amino acids, namely aspartic acid and glutamic acid, respectively, by adding of another amino group to each. The hydroxyl part of the acid is replaced by the N2 radical. Since amides contains more nitrogen than the amino acid, they are transported to another part of plant by xylem vessel. In addition, along with the transpiration stream, the nodules of some plants, example soybean, export the fixed nitrogen and urea. These components also have particularly high nitrogen to carbon ratio. The hydroxyl part of the acid is replaced by the NH2 radical. Since amides contain more nitrogen than the amino acids, they are transported to other parts of plant by xylem vessel. In addition, along with the transpiration stream, the nodule of some plants, example soybean, export the fixed nitrogen as ureas. Alright, now come to summary. Plants obtain their inorganic nutrients from air, water, and soil. Plants absorb a wide variety of mineral elements. Not all mineral elements that observe are required by the plants. Out of the more than 105 elements discovered so far, less than 21 are essential. Less than 21, that's 70, are essential and beneficial for normal plant and growth and development. The elements required in large quantities are called micronutrients, and those required in small quantities are micronutrients. These elements are essential constituents of protein, carbohydrates, fat, and acids, and they take part in various metabolic processes. Deficiency of each of these essential elements may lead to symptoms called deficiency, fluorescence, necrosis, and growth, enteric, etc. Are some prominent deficiency. Alright. Sensitive plants absorb minerals through roots by the passive processes. They are carried to all parts of organisms through xylem along with water transport. Nitrogen is very essential for the sustenance of life. Plants cannot use atmospheric nitrogen directly, but some of the plants are associated with nitrogen fixing materials, especially roots of legumes. Such can fix atmospheric nitrogen into the biological unstable forms. Nitrogen fixation requires a strong reducing agent and energy in the form of ATP. Nitrofixation is accomplished with the help of nitrogen fixing microbes, mainly sodium. The enzyme nitrogenase, which plays an important role in biological fixation, very sensitive to oxygen. Most of the processes take place in anaerobic environment. Energy to be required is provided by the respiration of host cell. Ammonia produced following infiltration is incorporated in the amino acids as amino group. Okay, done.